Hi everyone, whoever is watching right now. I don't know who are watching, but I can see the live chat. Oops, I have to mute my conversation there. But anyway, if you're here, we're going to first discuss. We were discussing about immunology today, and in these open classes, I at least want to give you more confidence and make you feel comfortable in this particular point, such that when you appear for the exams, for example JAM, JNU, or many other exams, you can really excel in that exam, right? So if you're in the live chat, feel free to chat with me, and as we go along, we're gonna discuss your questions and answers, right? But let me begin with the question summaries and the question frequencies. So there are a number of questions asked in the y-axis, and the year is given on the x-axis. So you can see every year at least 2 to 3 questions are asked from immunology section, right? Now immunology is not really taught well in your course, most of the courses at least. I don't know about everything, but what happens, that really gives you a little bit sort of less confidence in this particular subject, and you feel like, okay, I cannot do it, it's not possible for me to do it, or you just ignore the questions from there. But actually the questions that are asked from immunology is very very easy. So let's see how we can talk about immunology and all these things, and at least at the end of this course you would be able to get defined 2 marks from immunology sections, right? Because we're going to really discuss all the questions that appeared in previous year. We're going to discuss relevant concepts, explaining each of that in details, such that you know how to deal with these kind of concepts and questions. Okay, before that, I just want to tell you a detailed planning of immunology. So what happens is, the most questions that are asked from immunology is from antigen antibody interaction, which we are going to talk about today. Then a little bit about birth and the birth of the immune system, the components of adaptive and innate immune systems. So about the second point, we have already discussed it in the first lecture of this continuous series. And for the third, T cell and the B cell development, we're going to start today with the B cell development. And since antibodies are actually secreted by the mature B cell, the plasma cell, so we first decided to learn about a little bit development, their maturation, and then go to antibody structure. While we go to antibody structure, we would at least have the understanding about the context, that who is secreting them, right? So that's today's plan. But then there is an important concept known as hypersensitivity reactions. Hypersensitivity reactions are actually 4 kinds, type 1 to type 4, and they frequently ask questions from the hypersensitivity reaction. And then comes MHC genes and MHC processing. So these are the broad headings of the topics which are so important for IIT JAM. So, but anyway, for any exams you're going to sit, all of these concepts are important. So my target today is to make you feel more comfortable about all of these topics, and I would try to go as slow as possible, really slow, to tell you about these things. And as we go along, we would discuss all of these questions, keeping in mind how the IIT JAM question was asked, like at what point of which concepts they ask the questions. And if you really scan the questions, which I have already done, I know they ask questions which are really conceptual. So we need to boost our concepts at least at the end of this lecture. Before that, I would really think that after the end of these lecture, you would be feeling more confident about the concept. Then you would have a deeper understanding about the subject. You can plan better and you can really boost your analytical skill. I'm also available in an Unacademy Plus platform where we achieve milestones within 15 or 30 days. So our revision course is going on. Now, if you want to join there, you can always join anytime using my coupon code AP10 to get a 10% discount. But this is an open class for everyone who cannot afford that money, and also they have a dream to study in IITs or go and do their higher education. So this is for everyone, right? So without wasting further time, we should begin. Okay, so we are now going to talk about a little bit about B cell development, because that's our point of study for the first 30 minutes. Okay, so let's talk about B cell development. I don't have to introduce you to what is a B cell, but you might not be familiar with how B cell development takes place and how does it take place. Let's say you're reading this topic from a textbook. Instead of reading from the first page and to the last page of the chapter, I would tell you the way how you can read it, and I would also tell you the way how I did when I cleared these exams. So first, let's say if it is a B cell development, this topic known as B cell development, and I'm going to study about that. So what I'm going to do right now is first list down few of the questions. The questions are my framework of understanding. So first question, B cell development. Do I know about B cell? That's my first question. Okay, if yes, then fine. If not, then I would first focus a little bit of time to understand what is B cell. Then where does B cell come from? I mean, how they are born, where they develop, these developmental aspects. We would really understand the developmental aspects of that, right? That's the second question I would ask. Third question is like how the maturation process of the B cell take place. Where does it take place? How does it take place? These kind of questions. And then how B cell can secrete antibody, how other processes relevant to B cell take place, how other immune system actually communicate with B cell to make them do what they do. So these questions would be my learning goals while I'm reading a chapter. I'm not going to read the chapter from top to bottom and just like that. I mean, that is one of the way of reading. It gives you a perspective how the author wants to take you through this whole chapter. But here what you're doing is more or less what you want to do, and you're preparing for this particular exams, but at the same time you are ensuring that you don't miss out in some concept, and you don't want to just rectify it and vomit it in your exam copy, right? So as we go along, we would really learn about the concepts. Okay, so as we said, B cell development, it takes place in the bone marrow. Now bone marrow is present in the big bones such as your femur bone, let's say humerus, radius, etc. Now let's get a cross section of this bone. As you see in the cross section, there are specific regions which is having bone cells, let's call them osteocytes, and or osteoblast if they are immature, and they have blood vessels running inside the bone marrow. So you have a vascular component and a osteoblast component. You can name them a vascular niche and a endosteal niche. In the endosteal niche, that means near the bone cells, there are several cells which are known as hematopoietic pluripotent stem cells. These hematopoietic pluripotent stem cells are the mother of our whole immune system, as well as they are mother of our all type of blood cells. In simple words, our blood cell, our immune system, everything is born from hematopoietic pluripotent stem cell. Here I want to pause for a moment and tell you what does pluripotent stem cell mean. You might have heard the term totipotent, pluripotent, omnipotent, multipotent, and all of these things. So what they really mean? Pluripotent stem cells mean they are pretty much naive and they are pretty much immature. They don't have any constraints that you have to become this and that and that. So they have the possibilities to become anything. So what is a totipotent? Totipotent stem cell by definition can give rise to embryonic and extra embryonic tissues, both the tissues, okay? And an example is zygote. But a pluripotent stem cell are little bit more differentiated or a little bit more primed, I would say. So they can give rise to all the embryonic cells. And here for hematopoietic pluripotent stem cell, they can give rise to all the cells which are associated with the blood system or the blood system, right? So now you understand what is a hematopoietic pluripotent stem cell. We really break down the term one by one and try to understand what they are. Now what is going to happen is that the hematopoietic pluripotent stem cells can give rise to progenitors. Now progenitors are a little bit primed, so they have defined fate that they would either become this or become that. Now here is our endosteal niche, which I have already introduced you. So endosteal niche is the region or the locality, okay, where you see that they reside, like the hematopoietic pluripotent stem cell reside. So you live in a colony or a particular place, right, or apartment or a housing society. So you have a small society, right? So exactly these cells, they also have society. So this endosteal niche is a society where the hematopoietic pluripotent stem cell reside. Now as they mature and become more differentiated, they move towards a vascular niche or near the blood vessel, the society which is near the blood vessel. So imagine there are two colonies inside the bone marrow. One is near the blood vessels, one is near the endosteal cells. So they would migrate towards the vascular niche and get secreted into the bloodstream. From bloodstream, they can migrate to several regions. So let's talk about what type of progenitors hematopoietic pluripotent stem cell can give rise to. So it turns out hematopoietic pluripotent stem cells can give rise to two different kind of progenitors. One is the lymphoid progenitor. Two is the myeloid progenitor. Now lymphoid progenitor is the mother of whole adaptive immune system components. Your whole adaptive immune system is born from the lymphoid progenitor. Your myeloid progenitor, on the other hand side, is the mother of all the components of the innate immune system and your other blood cell types, such as your RBC, your platelets, etc. So now at least this particular slide is very important because they have asked questions that, okay, here are some cell types. These cell types are born from your lymphoid progenitor or myeloid progenitor. So this is a frequently asked questions, and this concept is important, that which cell are born from which type of progenitor. But isn't it interesting? From one particular common lymphoid progenitor, you have both T cells and B cells, and also from myeloid progenitor, you have so many different type of blood cells such as neutrophils, basophils, mast cells, etc. So how this work at the molecular level? How does lineage commitment take place? These are very important questions you should ask yourselves to understand this system better, right? Okay, so let's focus on the lymphoid progenitor for a moment, and let's just forget the myeloid progenitor for a bit. We would come back to myeloid progenitor later on. So let's see what are the cell types that lymphoid progenitor or the common lymphoid progenitor can give rise to. It can give rise to T cell precursor, it can give rise to B cell precursor. So the precursor is a vague terminology, I would say. So they're ultra immature at this stage. They cannot do what they want to do, even if their lineages are not committed to, to a certain extent. Other than that, there are third type of cells which are produced by the lymphoid progenitor, which is natural killer cells. But anyway, our focus today would be the B cell precursor and how they develop inside the bone marrow. Okay, so now we are inside the bone marrow. As I have already discussed, that there are so many osteoblast and bone marrow stromatal cells, which are supportive cells present in the endosteal niche, and there is another niche which is near to the blood vessel vascular niche. Anyway, these moral of the story is these stromatal cells actually help in the maturation, or they give the support for a very mature B cell to develop. So next 20 minutes we are going to focus on that how these stromatal cells can really help these immature B cells to develop, right? So first of all, the stroma comes from a Greek word known as matters. Now stroma is a substratum, okay? So these cells are very large, they're adherent cells, and their main job is to nourish the growth of the immature B cell and help them to develop. So stromatal cells, like any other substratum, hold different B cell stages. Now these nomenclatures, these pre-B cells, pro-B cells, these are a bit confusing, but imagine, let's say we talk about your childhood. So let's say when you are like 5 years old, you used to get called by one name, let's say kiddo, and then you became teenager at your teenage, and now you become 20s, right? So you have this whole process of your lifespan. So this is the same B cell which is undergoing a maturation at several time points. Okay, so we understand. So yeah, so yeah, hi Mitan. Okay, hi, I can see you. I hope you can see my podcast as well. Yeah, you have written so many highs. I don't know, it's a glitch in the system or anything. I can see you, Mitan. Anyway, so we are going to talk about next that how the B cell is supported by these stromal cells. So from hematopoietic pluripotent stem cell, the first cell type which is produced is pre-pro B cell. Okay, so hi Alok as well, so I can also see you. I mean, this is my very initial streams, so I'm a bit nervous about it, but I'm also under stress like you guys who are going to give the JAM exam. So yeah, let's start and talk about it. So what our goal today is, sit in the lecture for 90 minutes, at least take 2 secure marks for your IIT JAM. And how it is going to be done, we are talking about immunology. While you rewind, you can get the idea what we are going to talk about. But we are going to talk about first B cell development and maturation, then using that as a link to the next concept, we are going to learn about antibody structure, because B cells secrete antibody, one version of the B cell which is plasma cell. And then we are going to learn about the antibody antigen interaction and how the antibody interaction is used in several immune techniques. So that's the total learning goal today. While we go forward in these conceptual roadmap, we are going to also look at the questions that are asked and what are the concepts that you really need to focus for IIT JAM. So let's see. When you rewind this video, you would also get the detailed plan in the front. But right now, we're inside the bone marrow. Here, our bone marrow stromatal cells are supporting pre-pro B cells. Okay? Now, pre-pro B cells are marked by B2220 receptor. Okay? Yeah. So, okay. So you're saying that I'm too fast. Okay, I'm going really slow. So B220 is actually a lineage marker for a pre-pro B cell. It has all the adhesion molecules, but you don't really need to understand all these bloody details. What you really need to understand is they have adhesion molecules by which they can actually interact with the stromatal cells. Now, okay, here is our stromatal cell, which is expressing an adhesion molecule known as VCAM1. Again, the name is not important, but you have to know that the stromatal cell express some molecules which work like an anchor for the immature B cells. Now, the pre-pro B cell also have a molecule complementary to that, VLA4. Now, what happens is VLA4 interacts with VCAM1, and this interaction is very important. That tethers the B cells or the pre-pro B cells into the niche of the stromatal cells. Okay, it leads to c-KIT expression onto the pre-pro B cells. c-KIT is super important for B cell development, I must say. Now, next stage, what happens? There are other interactions like c-KIT and SCF interaction. So one interaction lead to another interaction. This is a common theme. Now, stromatal cell also secretes several chemokines, in this case CXCL12, that help the pre-pro B cell to mature. How? So B cell has to create its B cell receptor, right? B cell receptor is antibody, a membrane bound IgM, right? Okay, so a membrane bound antibody. So first of all, that antibody need to undergo VDJ recombination. And VDJ recombination we are going to cover today. But just to give you a context, VDJ recombination is going inside the nucleus because the genetic material is present in the nucleus. So VDJ recombination you cannot imagine isolatedly. It is going on inside the pre-pro B cell at this particular stage. And these chemokines, which is secreted by the stromatal cells, really help the pre-pro B cell to undergo VDJ recombination. VDJ recombination is a super important concept you should know about, know while you're going to appear in IIT JAM, JNU, or many exams. So it's an important concept. Okay, so in that stage, you have c-KIT and SCF interaction, and CXCL12 works like a very important cytokine, which allow the B cell to survive, proliferate, and move forward to the next stage, right? Okay, at this particular stage, the DJ recombination takes place, right? Okay, so now we move on to the next stage. Now they have detached from that particular stromal cell which was looking at light brown in color. Now this is a different stromatal cell. Okay, so give me a second. I just need to reply something. So these stromatal cells are different. How they are different? They are secreting IL-7, okay? Sorry, they are secreting IL-7. This IL-7 is very important cytokine because somebody need to tell you that, okay, now you should finish your VDJ recombination. So VDJ recombination is a complex process, requires a hell lot of energy. B cell won't do VDJ recombination all the time, right? So what is the triggering factor for VDJ recombination? This is this particular interleukin, and that is secreted by what? Secreted by the stromatal cell, and it is received by the recipient, which is the early pro B cell. Remember, the pre-pro B cell now became a bit more mature because it has undergone the DJ recombination. Now it has the ability to go VDJ recombination, because VDJ recombination, when we talk, we will know that it would take place in a sequential fashion, first DJ and then VDJ. Okay, I hope you guys can follow who are online. Anyway, at this particular stage, several transcription factors such as PAX5, which you don't need to remember the name, you just need to know that some transcription factor at this point help to do the VDJ recombination. Since we are talking about it, we learn this process in a bit more details. Yeah. So I don't know people who are online, or somebody is asking me it is paid or free. So this particular lecture is totally free. It's YouTube online lecture, right? Okay, so here whatever you can see is the IL-7 receptor, which works in a JAK-STAT signaling pathway. So here is our IL-7 binding on the surface. It leads to STAT activation. Now the STAT would express many genes, but the common theme is it would make all the genes which are essential for VDJ recombination. So all of these things tells you that VDJ recombination is a very important topic or very important life event in case of a B cell. Okay, so we know that there are several factors which are augmenting these VDJ recombination processes, several transcription factors such as EBF1, E2A, PAX5, etc. Okay, now this is pretty important, right? But once, at the end of their interaction with the stromatal cells, they would actually undergo rearrangement of the heavy chain of the antibody. Now, you don't have to really focus right now because we are going to talk about antibodies, heavy chain, light chain in more details. So for now, they have a heavy chain, but they don't have a proper light chain. So after this point, the light chain recombination would take place and they would be secreted out of the bone marrow. To where? To the secondary lymphoid organs. The secondary lymphoid organ in this case is mostly lymph node, or otherwise they would be transported to spleen. So lymph node, you can imagine that to be an army base camp. So they are ready to be deployed to the border and fight, but before that they need to undergo the training and maturation, right? So the training phase is happening for the B cell in the bone marrow, and at an immature state they are going to the lymph node. Now in the lymph node they would get practical training. That means, okay, the combat experience. That means, okay, what type of pathogen might infect us, or what kind of pathogenic antigens are there. So all of these would be recognized in lymphoid, secondary lymphoid organs such as lymph nodes. You guys can still follow, guys who are like present here right now, and give me some indication in between just to make sure that you guys are with me. Anyway, So inside the lymph node, which we call as our army base camp, they have specific areas for residing of the B cell and the T cell. The area where B cell resides, we call it germinal center, or otherwise they reside in a specific area which is a dense structure known as germinal center. Okay, so we learned how B cell develops. Now we should learn how B cell gets activated. So activation, excitation always requires activation energy. So you cannot get activated always without any motivation, right? So who is motivating the B cell to get activated or secrete antibody? So that's our first question that should strike in our mind. Okay, so you already know that the B cell are developed from the lymphoid progenitor. Just a refresher, quick refresher. And inside the bone marrow, in a subsequent way, the stomatal cell is allowing the B cell development, allowing the VDJ recombination. Then it is secreted into the bloodstream. From where? From the bone marrow. And now in the bloodstream, they would go to other lymphoid cells. T cells are also secreted from the same progenitor, okay? They're also secreted into the bloodstream. T cell goes to the thymus, whereas B cell goes to the lymph node or spleen. So following these particular B cell, which is now going to the lymph node. Okay, they moved inside the lymph node. Now here you have a particular lymph node structure. We zoom into it and look at the anatomical location for 2 minutes because it's important to understand. You have germinal centers or follicles. We should say germinal center is a dense center where there are a hell lot of proliferating B cells. Now the lymphoid follicle is present in the lymph nodes. There are also cortex, medulla in light blue and pink, and also there are cortex. This dark green region is known as the paracortex. Okay. So now what is important is inside the lymphoid follicles, what happens is very important. So inside the lymphoid follicle, what you're going to do is you're going to proliferate your B cells. Wait a second. Something gone wrong. We are back again. Okay, so always follow my laser pointer. Here you can see, here is an immature B cell. So it has its B cell receptor. Now it is capable of detecting, but it's not yet trained to detect. So what it's going to do first is going to interact with several other cell types. Let's see what cell types. So first, they can clonally expand, they can get proliferated. Otherwise, they can be immediately differentiated to a cell type. Okay, this proliferation phenotype is important because once they are proliferated, they can either become a memory B cell or they can be becoming a plasma cell from there. So these are two alternative outcomes, right? Now let's see how the first step, the activation step, takes place. So when we talk about activation, there are two important concepts: a TH dependent activation, which requires the guidance from the T cell. Okay, this is a one mode of activation which requires guidance from T cell. Other mode is known as TH independent, which doesn't require a guidance from T cell. So there are specific molecules on the T cell which are present, which are interacting with the B cells, help them to give intracellular signals, and all of these signals tell the B cell now it is the time you should really mature and differentiate into antibody secreting plasma cells. And many other chains takes place in the B cell, such as somatic hypermutation, such as class switching. That means by default they have membrane bound IgM. Okay, but this is not the prevalent antibody. There are many other antibodies that are produced by the B cells. We know, right? Hi, so Nilanjana is actually online. So hi Nilanjana. It's also my first time YouTube streaming, live streaming. But anyway, so here you have the B cells which are getting the influence from the T cells. Hi Alok. So now the T cells, when they communicate with these B cells, they would give them the necessary signals such that the B cell would mature into a plasma cell. The other mode of activation is by a long repetitive antigen which might be a part of a bacteria, or let's say a part which is present in the bacterial flagella. So it involves the TLR signaling and also a signaling from your BCR. Okay, or many of your membrane bound BCR receptors could be cross-linked by a repetitive antigen which is derived from bacteria or many other species, let's say. So these are, but whatever you can see, they doesn't require the TH dependent, I mean they doesn't require the help from the T cells, right? So this is T independent antigens. So T independent antigens are mostly bacterial flagella components, many other components of the bacteria which are repetitive in nature, which would cluster or cross-link B cell receptors and allow their activation. Other antigens which are T dependent, they would be first displayed to T cell, and the T cell would understand, okay, this is something wrong, so I should tell B cell that now you should activate. So this is the concept behind T dependent and T independent antigen, which is very important in terms of IIT JAM and many other examination. So let's say the flagellin, which is actually the protein which builds up the flagella of the bacteria, or let's say the LPS, which is a component of the membrane, right, lipopolysaccharide. This is actually specific signatures or specific antigens which are T independent. Now, be it T dependent activation or be it T independent activation, both the cases, what happens when it is activated, the B cell is activated, they would really enter the cell cycle. Now you know what happens when it enters the cell cycle. It would divide, it would start divide rapidly and proliferate, increase their number, and all of these thing is happening in your follicles, right? The lymphoid follicles where they would form the germinal center. Now this interaction between T cell and B cell is taking place in the paracortex. Paracortex, you can imagine it's a T cell area, and after the interaction they would be differentiated into plasma cells. These plasma cells would quickly secrete antibodies, mostly IgM type, because IgM is the default state of antibody, so it would quickly secrete it into the bloodstream. So from the bloodstream it would reach your whole body and go to the place possibly where the infection has happened. Now the B cell can also proliferate. I mean, instead of giving rise to the plasma cell, the other fate is they first proliferate a lot and then give rise to plasma cell, because they really always want to increase their numbers and then proliferate. And some of them can also become a memory B cell. Okay, so when we learn about secondary immune response, that is so important. So let's look at the process, what is happening inside. So this is the enlarged view of the germinal center. If you remember, germinal center is very similar to an army barrack where the B cells are residing. Let's say the land troopers are residing, and the T cell area is a very similar region where the, let's say, the navies are residing. So these are two different regions. Now here in the germinal center there is a trainer, there is a coach known as follicular dendritic cell, and there they undergo many different processes. Either they can recognize antigens. If they can recognize antigens which are present, I mean displayed on follicular dendritic cells, then what happens? They would survive. Otherwise they would die. They would die by apoptosis if they are not able to recognize it, or they are not able to find the or they are not able to interact with a high affinity. They would die. There could be second possibility. Okay, so the second possibility is like this follicular dendritic cell, they can allow receptor editing and making some receptors which are of very high affinity and allow these things to pause and then give the B cell a chance to survive. There is also a possibility, like, so I mean the low affinity problem could be rectified by this correction method or fail-safe method, and if it is rectified, then why not? They can become the affinity matured plasma cells, which is the antibody factory. Okay, I hope this is understandable. If you think that this is not understandable, I'm very fast, you can tell me to repeat this part or that part of that, these things. Okay, anyway, so yeah, we are going to now talk about the germinal center. Inside the germinal center, something randomly happens in the genome, and this is restricted to the variable region of the B cell receptor genes. This process is known as somatic hypermutation. Somatic hypermutation can give rise to many different affinities of the B cell receptors, low affinity or high affinity. High affinity ones would be selected, low affinity ones would be get rid of, right? And all these mechanisms exist because body want to create diverse set of response against pathogen, because pathogens are always evolving. Their evolution is way faster than the human evolution, so they are devising ways each day how they can cheat the immune system and survive inside the body. And our body is always in a constant battle of producing counter mechanisms by which it can really develop its own immune system and can be able to perform a diverse set of response. So that is the constant battle between the immune system and uh, our own immune system and the viruses, okay, and the bacteria. Okay, so let's say these cells which are not undergoing, which has very low affinity, they would die. Which have very high affinity, they would survive, and they would become plasma cell. By this time, they would also undergo a process called class switching. So class switching means, okay, by default state, you would only produce IgM type of antibody. Now class switching ensures that not only you can produce IgM, you can also produce other isotypes. What is isotype? I haven't talked about it. I'll be talking about it, so stay tuned. Okay, so we pretty much learned about how B cell development takes place inside the bone marrow and inside the lymph node, how their maturation process takes place. Okay, so now that gives us a good primer to talk about the antigen antibody interaction, because antigen is secreted by the plasma cells, right? Okay, so now we are transiting to a molecular level. So we were at a cellular level of understanding previously, which was your adaptive immune system component B cells. Now we are moving our focus or shifting our focus to the antibodies, which are the secretory elements or sometimes membrane mount in the B cell. So let's look at it. So how did it all happen? It turns out it begins at 1939 when people get a faint idea about antibody. So E.A. Kabat did an experiment. They injected a particular antigen in a rabbit's body and then took out, so it was biochemistry era, they centrifuged it and took out the serum and then ran the serum in a gel, and they found few bands. Okay, these bands are known as alpha, beta, gamma. That's how they really put the nomenclature of. But what is important is antibodies are found in that gamma globulin fraction. So this gamma globulin fraction is corresponding to the antibody. Okay, so that is important to know from a point of view, from an exam point of view. Now let's talk about the antigen antibody interaction, right? So antigen antibody interaction takes place here. So these are the antigen binding group on the antibodies. Before we go to the structure, we should understand what are the possible molecular mechanisms by which they interact to each other. All of these molecular mechanisms that are listed here in this particular text you need to know because this was asked in one particular IIT JAM. But what is the common theme? All of these interactions are transient. Antigen does not want to interact with the antibody for rest of his life, though these interactions are strong, but these interactions individually these interactions are weak. If you talk about one hydrogen bond, it is way weaker than a covalent bond, but there are multiple small weak interactions which lead to the antigen antibody binding. So all of these interactions are present, are non-covalent in nature. There is no covalent interaction. I repeat, there is no covalent interaction between antigen and the antibody in the antigen binding groove of antibody. So this is an important question that people really asked in one exam. So we just quickly, since we are talking about this, so as we are progressing bit by bit, we should get the numbers. So this is a hot topic, so I'll show you the evidence. So here we have the last year question, and I'll show you where they have asked this question. Okay, look at this particular question. Antibody binds to, it's probably 2015, antibody binds to antigen in the solution through, it's a MSQ question, So they have given bunch of these responses: ionic interaction, van der Waals interaction, hydrogen bond, hydrophobic interaction, all of these. Actually, the correct answer is all of these possible interactions are present between an antigen and antibody. So as we moved along, we really need to understand, okay, this particular concept we should keep in our head, that they interact by non-covalent interaction, and no covalent interaction is present between an antigen and antibody. Okay, in the previous lecture we have talked about a little bit about all the cell types of immune systems. In the next question they said gene rearrangement is taking place in the following cell types. So we haven't even get to the point where we can talk about the gene rearrangement, VDJ recombination. So VDJ recombination is so important, we're going to talk about it. Okay, but looking at this question, you can understand these are the points, or these are the particular concepts you need to focus. Getting back to our own discussion, so we learned that what are the important interactions between antigen and antibody. Okay, now let's look at the antigen structure, antibody structure in a bit, and try to understand. So basically you have two chains. One is a heavy chain, which is roughly having a 50 kilodalton molecular weight, and a light chain which has a 25 kilodalton weight. So this thing which is marked in yellow is nothing but your heavy chain. It has an N terminus and C terminus. Note, the N terminus is the one which bind to the antigen. Okay, the N terminus region is the antigen binding site. So this is important information. N terminus region is the antigen binding site. Okay, and they have asked one question in one particular year, that okay, these N terminal region binds to the antibody, or C terminal region. We'll get to that. But now you have in between, and we are taking an example of very simple antibody, which is a IgG subtype. We are going to talk about other subtype as well, other isotypes as well, allotypes as well, and idiotypes, and get to know what these things mean slowly. So there are two basic fragments. One is FAB fragment. FAB fragment, remember, always have the antigen binding groups. So you just put FAB fragment, it can recognize antigen. You just put the whole antibody, it can also recognize the antigen because it has the FAB. FC region is another important region, because FC region is basically the region where several cell types such as macrophages, receptor, which are present in the macrophages, can recognize this FC region. Not only macrophages, neutrophils, natural killer cells, even mast cells, they have this FC receptor. So the FC receptor is so important. Other than that, you have disulfide linkage which are connecting each other. Okay, so I'm drinking a little bit of water and getting back to the lecture. So at least this particular slide might give you an important answer to some questions. Let's again juggle between the questions of years. See, we go to the question paper again. See here, people have asked that the antigen binding site in the immunoglobulin IgG is present at variable region of the heavy chain, variable region of the light chain, constant region of the heavy chain, and variable region of the heavy and light chain. So you can clearly understand this is the answer. We have already seen that in our lecture, right? So in our lecture we have seen the variable region is present in the heavy chain as well and the light chain. So both the chain collectively form the antigen binding groove. So this is the simple concept that they are trying to ask. It's not difficult, you can clearly understand. So you just need to understand the concept. And don't worry, this concept is asked in several years. So I'll give you another example. So 2015 itself, in other, maybe that question was 2017 or something. This question is 2015, where they say antigen binding site of an antibody molecule consists of N-terminal domain of the heavy chain, C-terminal domain of the light chain. So they are only trying to say that, okay, this binding groove for antigen is present where? N-terminus or the C-terminus? Actually the answer is N-terminus, right? Sorry, So the answer is actually N-terminus, so the C is the proper answer. But what you can clearly understand, this particular concept is a hot shot or a hot spot for their asking questions. So you really need to understand this particular concept such that you can answer the questions. So I hope you guys are still with me. Okay, now we move on a little bit. Now we talk about polyclonal antibody. What are polyclonal antibodies and what are monoclonal antibodies? Two terminologies might not be familiar, you guys might not be familiar. So let's talk about what are these. So polyclonal antibodies are antibodies which determines multiple different epitopes. Again, one terminology. What are epitopes? So epitopes are actually specific regions present on the antigen which is responsible for binding to an antibody. So the entire region does not bind to an antibody. The epitope only binds, and the region that binds to the epitope is known as paratope. This black antibody in the first picture can detect all the possible epitopes present in one protein. It is detecting one protein specifically, but all possible epitopes. But here in the second one, it only determines the blue one, not the green, not the orange, or not the red. So that means even if other epitopes are present, it is going to determine only one epitope. And the type of antibody we are going to talk about in this situation is monoclonal antibody. Monoclonal antibody determines single epitope. They are more specific than the polyclonal antibody. Okay, fine. Now we're going to talk about the isotypes. What are isotypes? So we have already we pretty much know. So yeah, hi Shudipto. Yep. So we pretty much know that antibody exists in IgG, D, E, A, and M, right? Actually, this variation, this subgrouping is known or termed as isotypes. So let me tell you what is the basis of this difference. Actually, we have already talked about the isotypes. I mean, we have already talked about the structures, right? So we have learned that there is a heavy chain and the light chain. In this case, all the green chains are the heavy and the blue chains are the light chains. And they are proteins, right? So they have to be encoded in the genome or the genes. So heavy chain has different genes. For example, if we talk about IgG antibody, it has a gamma gene, gamma heavy chain gene, or mu gene, or alpha or epsilon genes. So the genes are actually different. So they frequently ask questions like, okay, gamma, I mean, IgG subtype is encoded by gamma heavy chain or mu heavy chain or alpha heavy chain. So give the proper answer, something like that. And the light chain is pretty much similar for all of them, either lambda or kappa. So they have two options for the light chain, either lambda or kappa. So be the IgG antibody, it can have either lambda or kappa or even a mixed population of both, all of these things. So this is called isotype. We come to isotype more, but just want you guys to understand that the antibodies, when we talk about the antibodies on the surface of the B cell or B cell receptors, those antibodies don't have secretory function. So those antibodies are stuck on the membrane. How does antibody can get stuck on the membrane? But you say in blood, in serum, there are antibodies. So what are the differences between the secreted and the membrane bound antibodies? So let me tell you who secretes the antibody. Plasma cells, right? Not exactly B cells. Just like you are very different person at your 20s than your teenage. So plasma cell is, let's say, a B cell at its 20s. So plasma cell is a differentiated version of the B cell. Now the secretory antibodies don't have the transmembrane domain or the sequence coding for transmembrane domain. So how does that happen? It turns out when the mRNA is produced, in case of the membrane bound versus the secretory antibody, the secretory antibody would undergo a polyadenylation site choice, and that would form a truncated antibody, okay? And that would be really easily secreted, not tethered into the membrane. Okay, now very important question, very important concept for IIT JAM. They would always ask, okay, now we digest our antibody with pepsin or several other enzymes, and what would be the fate? How many band we are going to get in the protein gel, let's say. So here you can see there is a pepsin digestion, okay? So pepsin digestion really cleaves the Fc fragment and destroys it. So you would have the FAV fragment. So important, pepsin digestion give rise to only the FAV fragments, okay? Now if you treat with beta-mercaptoethanol or any reducing agents, what would happen? These disulfide links which are present in between the chains, they would get reduced. They would form the sulfhydryl groups, and as a result, what would happen? The interaction between these chains are broken down, the structure is broken down. But in the gel, how many bands you are going to see? You are going to see two bands, one for both of these heavy chain, one for both of these light chain. See, let's take a look that whether they really ask this or not. So let's just discuss that. In 2016, they asked this same question. See this question, which is written as question number 1. A pure IgG antibody was run on a SDS page under reducing condition. Important topic, reducing condition. So what does reducing condition means? Either they have treated dithioethritol or they have treated any reducing agent which would lead the breakdown of the disulfide linkage. So how many bands you are going to get in a gel stained with Kumasi blue, which is basically a SDS page gel. So what you're going to see is two bands, right? One band corresponding to two heavy chains, one band corresponding to two light chains. Okay, so this particular concept is asked in the exam, so this is important. So yeah, so let's get back to our concept. Fine. So when people looked at that there are so many different antigen types and there are so many different antibodies against them, so if we talk about the whole protein structure, which part is the most variable region? And it turns out inside the heavy chain and the light chain, in the variable region, there are small portions such as loops which are highly polymorphic or highly variable in terms of sequence features. That gives the scientist a clue that okay, these regions are so variable because each time you can generate a variable antibody or different antibody by changing a little bit amino acid sequence in this region, because these regions form the peptide binding groove or the antigen binding groove, right? So that's why it's legitimate for them to become more variable. And MHC, when we talk about MHC genes, we are going to talk about more about this thing. Okay, so hypervariable loop exactly give a loop-like structure which binds to the anti chains. And now we're going to talk about what are the functions of the antibodies. Out of many functions, they can really coat the bacterial surface with several antibodies. This is called attenuation, or like inhibition of the bacteria. So with so much coating of antibody on the surface, the bacteria really cannot do a lot of things. Moreover, there are many cells in the immune system such as neutrophil, macrophages, eosinophils, NK cells. All of these has the FC receptor here, the FC receptor. The FC receptor stands for fragment crystallizable, which is the tail of the antibody that is detected by this receptor. And once it is detected, what happens? What happens is these cells are engulfing the pathogen. So this is one of the way by your immune system can get rid of your bacteria in an antibody dependent fashion. Okay, not only antibodies can neutralize pathogen, they can also create hypersensitive response or too much overactivation of the immune system, and that is the case of allergy. IgE antibody is specially associated with allergy. You need to know this. And IgE antibody is the lowest abundant antibody in our body, and it binds to the FC region present in the mast cells. What it does to the mast cell? In the first exposure, it would make the mast cell sensitize, make a hell lot of granules filled up with histamine. But when there is a second exposure, mast cells would degranulate. And this degranulation allows the release of histamine. And what this histamine does is a variety of things, which are mostly harmful and irritation for our body. But these gestures are actually done to protect our body. But instead, if it is done in a uncontrolled fashion, it would be harmful for our body. So it leads to smooth muscle constriction, vasodilation, bronchoconstriction, and also glandular secretion. Imagine, let's say your body want to secrete more glandular fluid, let's say gastric HCl. So it ensures, okay, what are the pathogens present in our GI tract would be dead. But if it try to secrete a hell lot of glandular materials such as a hell lot of HCl, it could be also damaging your own mucous epithelia, right? Moral of the story, you need to know IgE is important for an allergic response, and allergic response are also known as hypersensitivity response. Okay, not only biological aspects, antibodies are used in several techniques in biomedical research, such as ELISA, such as Western blotting, immunofluorescence, chromatin immunoprecipitations, and many more. So antibodies are really useful for your research purposes. Okay, so let's talk about how antibodies are really useful in terms of biomedical research. Antibodies could work like medicine. You would be amazed to know that the breast cancer, which is happening due to HER2 mutation, which is a mutation in the EGF receptor, which is a growth factor receptor, it leads to hyperactivation of the growth signaling pathway and allows a tumor mass to be formed in the breast. But there is an antibody which is called HER2/neu, which is against this receptor, which can block this constitutive active state of the receptor. Not only that, some of the allergic medication which is not antihistamine type utilize the antigen antibody interaction. How do they do it? So the way they do it is like they basically bind to the Fc region of the IgE. Now, if IgE's Fc region is blocked, it cannot bind to the mast cells and it cannot show its harmful consequences. So that is how antigen antibody interaction is beneficial to study in context of biomedical research. It could be applied as a medicine, right? Now, if you guys are still here or you guys are not falling asleep, you guys can watch this lecture in segments. But now we are going to talk about the 3 important terminologies. These are isotype, allotype, and idiotype. So let's see what they are. So first, isotype. We already talked about isotypic variant means the variants of heavy chain and the light chain which are encoded by several genes. So we have looked at that there are like specific heavy chain genes, right? For IgG it was gamma, for IgM it was mu, etc. So the point of difference is basically the heavy chain and the light chain gene actually. So these are like the genetic variants. Now we have IgA, D, E, M, and IgG present in our bloodstream. Now let's look at a little bit about these isotypes to understand what are the important concepts you need to know. First of all, this isotype differs in sequence and number of constant region domains and the hinge structures. Okay, so their structural point of view, they are different. For example, IgM has very different structure than IgD, right? And they are also encoded by different constant region gene segments. Okay, so we would talk about all these isotypes and what are their function. First, let's talk about IgG. IgG is the most abundant antibody present in our body. It can transfer via placenta and get to the embryo. IgE is super important for allergic response, right? Now then, for classical complementation pathway, IgG or IgM is important and the whole slide is important for you. Whereas IgA is a key secreting variant, okay? So different isotype has different function, different abundance, which we would be talking in details in a bit. But let me tell you that allotypes are actually allelic variants. What are allelic variants? So let me give you a very cool example. So we know we have an Android phone, right? We have an Android phone. This Android phone has, let's say right now, KitKat, right? It's a smartphone. It has a KitKat operating system right now. But let's say other smartphone which has a smart operating system, Jelly Bean or Marshmallow. So there are different versions, right? But the thing is same. So allelic versions, alleles are the versions of the genes, right? So the allelic version would give rise to allotypes. So let's see allotypes, what are these things. So heavy chains and the light chain genes have several gene segments, and these gene segments actually are different in allotypes. And how? Because they are different in different type of alleles. So both the cases, it might have a minute difference in the allelic variation that would lead to a small change. For example, here you can see there is a red domain and there is a yellow domain. That's the point of difference, the minute difference. But this minute difference could have a huge effect in terms of affinity, because if you have this minute difference, one antibody might have a very high affinity towards a particular pathogen antigen, or one might not have a very good sort of affinity. Okay, so that is pretty important. Now we are going to talk about the nomenclature, how the nomenclature takes place. I mean, they are termed as IgG, then M1. Okay, so the kappa chain M1 and G for gamma chain is like that. So that is why their allotypic variants are named. So in the second one, they have the same gamma chain but the different kappa chain. Okay, and actually if you see the sequence information, you would see they really differ from each other with a very small sequence features. And these sequence features, though it look like very small, but they could have very big consequences in terms of their affinity. Idiotypes, what are idiotypes? Idiotypes are same same antibody class against different antigen. So both these antibodies which are shown in the screen are actually IgG type. So their isotype is IgG, but they are against different antigen. Here a blue antigen, here a green antigen. So definitely their binding constant, their affinity, their avidity, they are very different, right? So that is how they are known as idiotypes. So IgG against antigen A or IgG against antigen B, their properties of affinity, avidity is very different, right? Mostly the difference lies in the hypervariable region, which is the point of difference. Okay, now we go back to our isotypes and try to understand which isotype is important for which biological functions, and we're going to cover only those topics which are super important, not going to talk about anything which are not important. Okay, we're going to first talk about IgG. IgG is mostly 90%, 75% of the whole antibody population, and this is a super abundant antibody, most abundant antibody. It can cross the placenta, which is a very important information you need to know. It has capability of fixing complement pathway. Maybe we are going to discuss about the complement pathway today. And then it can fix complements and allow the innate immune system. Now, membrane-bound IgM is very different from the secreted form of IgM. Secreted form of IgM is a pentamer with a joining chain here. Now, if they ask you in the examination that, okay, if I really reduce this IgM antibody with a reducing condition and then run it on a gel, how many bands you are going to see? The answer is 3. Because see here, you are going to get a hell lot of bands which are corresponding to the heavy chain. One type of band would be corresponding to the light chain, and one last band would be corresponding to this yellow J chain. Okay, so this is a popular question that they can ask. Okay, the membrane-bound antibody, we learned that it is different from a secretory antibody because the secretory antibody doesn't have the transmembrane region, and that's how they are different. That's why they are not tethered, they're secreted. Very important concept right now. The very important concept is the primary and the secondary immune response. Okay, the primary and the secondary immune response. So first of all, let's say you get an antigen exposure some point of your time. Now what happens is, like, there would be some lag. So if you have an antigen exposure, let me take a pen. So here you have an antigen exposure. Let's call it day 0. So there is a 10-day lag till we see the antibody. Why there is a lag? The lag is there because of one reason. Because whenever you infect your body with a pathogen, that doesn't mean the adaptive immune systems would be then and there activated. The innate immune system components need to recognize them. They need to go to the army base camp, which is the lymph node. Then they have to interact with the T cells and the B cells. The T cells would then activate the B cells. B cells would undergo further processing. Then only the antibodies could be secreted. Phew. The thing is, the first thing that is secreted by the B cell is IgM type of antibody, because as a default state, the IgM is a default membrane receptor. It is default state, it is default expressed. But even if you wait a little bit more, the response is actually IgG based. They're secreting more IgG antibody than IgM, but that's a later part. First IgM to appear and then IgG to appear. Okay, afterward what happened? After, when the pathogens are neutralized and sort of complements are fixed, cells are burst, the bacterial cells, then what would happen? What would happen is your antibody level in the serum would drop, because there are not too many circulating plasma cells or too much serum antibody, so the level would drop. But let's say after 2 months or 3 months you are getting a same pathogen infection, second exposure by the same antigen. Now what you would see, the lag phase is very small. Like within 1 or 2 days, IgM would be produced, and now there is a huge, at least 100-fold more IgG activity or IgG presence. So why the secondary immune response is faster than the primary immune response and way higher in the magnitude? The simple reason is, during the primary exposure, your B cells undergo a huge proliferation in the lymph node. Thus, most of them has died. After the work is done, many of them has died. But few of them lived. Few of them lived to do what? Few of them lived to do an important function, which is memory B cell. Now, memory B cell have a memory of the pathogenic antigen. Now, next time when a same pathogen enters our body, memory B cell hits. And memory B cell eliminates the pathogen way quicker than the primary immune response. Now, this is not very new these days. That's why people say if you have a event of pox or something like that, the next time you won't have the pox. It's not true that you won't have the pox, but you can also have the pox, but the immune response to that would be way quicker. Okay, so this is a very important concept about primary and the secondary immune response that they always ask. Yeah. Next, we talk about IgA antibody. Again, they can ask this question: if you digest IgA with, or let's say you treat IgA with a reducing condition, such as dithiotritol, or let's say what was the other compound? Beta-mercaptoethanol. So what would happen? Again, your heavy chains and the light chains would be separated. Heavy chain correspond to one band, light chain correspond to one band, and your J chain correspond to other band. So you would get 3 band. Regardless, IgA can comprise to like 15% of the antibodies, but it is the prevalent isotype which is found in bodily secretions such as breast milk, saliva, tears, mucus, etc. Okay, your urine don't have your IgA antibody. If you have a antibody in your urine, that means it's a pathological condition. It's not a normal situation. But most of your bodily secretion have IgA antibody as a prevalent isotype. This question was always asked in JNU JAM many times. Okay, so you guys are still with us. Can you guys please tell me which of you guys are still with me? You can always watch the repeat podcast, but you can always do the live chatting with me while we are running the session. These concepts that we are going to talk about in this class are super important for IIT JAM. Okay. Now, the IgA, mostly we imagine this kind of picture of IgA while we talk about IgA, but they can exist in dimer, tetramer, or even monomer. But mostly the secreted form is tetrameric, and they are joined by a J chain, okay? Now, they are secreted by a process known as transcytosis. So, transcytosis, by definition, is a process by which two-way transport happens, like endocytosis from one side, exocytosis from other side. So, the antigen gets in from one side, gets out from other side. This is called transcytosis. So let's see a process, or let's look at it into details, that how IgA antibody is secreted into the breast milk, because it is prevalently present in our colostrum, right, in the mother's colostrum, which the baby feeds on at the initial part of his life. So here is the cross section of the breast. You would see there are multiple mammary glands. These glandular cells would secrete milk, so they have milk inside them, but you have glandular epithelial cells, which are actually columnar epithelial cells. Now let's look at it. So breast doesn't have any source for antibodies, right? So antibodies would come from where? The nearby lymph node, and lymph nodes are dispersed all across your body. So yes, the antibodies are coming from nearby lymph node, but they bind to the poly-IgR receptor, which binds to the J chain of these IgA, and allowing the receptor mediated endocytosis. That means they engulf it from one side, and then they exocytosize the same thing from the other side, and the receptors are recycled. But the now antibody, the IgA antibody, is secreted into the breast milk. Now it would be secreted out of the breast, and it would go to the the newborn baby as colostrum, right? Okay, now we go a little bit further to talk about IgE. IgE is another subtype. What you really need to know about IgE is they are associated with allergy, and their abundance is very less. So they are lowest possible, lowest abundant antibody in the whole immune system, in your serum maybe. Now some people have a higher level of IgE. They are more susceptible to atopic. I'm pretty much sure you have heard about this evidence that, okay, I'm lactose intolerant, I'm intolerant to this food, that food, gluten intolerance, etc. So this actually is based on too much IgE present in their body, and they are known as atopic individuals. Okay, but mostly IgE is a prevalent isoform which is important for allergic response or type 1 hypersensitivity response. If time persists, we're going to look at hypersensitive response. If not, we'll look at it in another class. Okay, let's say you're going through a park and you have inhaled an unknown pollen grain which is shredded from a particular tree. Okay, it happened to me many times. And now this is inside your nose. Inside your nose you have nasal epithelium, these gray cells, and you have this mucus, this slimy looking blue stuff. And inside that you have your immune cells. Inside the epithelia you have your, the inner side of the epithelia, you have your immune cells such as macrophages, dendritic cells, B cells, etc. So what happens is, whenever these pollen grains or these antigens, which are treated as antigens, come in, these B cells, which are antigen presenting cells as well, recognize that and activate the T cells. Now, of these B cells, they can also get activated and forming plasma cells secreting IgE antibodies. Now IgE antibody can bind to the mast cells, sensitize it, and the second exposure it can really degranulate the mast cells, and that is how the degranulation of the mast cells is causing many symptoms of the allergic reaction, such as glandular secretion, redness, vasodilation. Vasodilation would allow increased inflammation, smooth muscle constriction, so you might have a breathing problem because you have bronchoconstriction as well, bronchial spasms. Okay, we have talked enough about isotypes, allotypes, idiotypes, their function, their structure, all of these, but we skipped one important thing. The important thing is the kinetics of antigen antibody interaction. This is very simple. Antigen binds to antibody in an antigen binding groove, and the interaction types are all sort of non-covalent interaction. So let's say we try to write it in format of an equation, okay, and try to derive and try to get a sense that what is the strength of this interaction. So generally, for enzyme substrate reaction, or let's say receptor ligand interaction, or let's say antigen antibody reaction, you write the reaction in a simplified manner, like antigen binds to antibody forming a complex. So if the interaction is very strong, only the forward reaction is preferred. If the interaction is very weak, so the dissociation part, that means the backward reaction is favored. And you can name the forward reaction constant as a K association constant, which simply means K on by K off. And the backward direction can be thought about like the dissociation, which is a ratio of K off by K on, right? Now this is super important because they asked a question like this. I'll show you this question in a moment. It was a numerical answer type question. So where people said the same equation they have given and then say, okay, what is the value of the dissociation constant, right? And what they have given, K1, that means K on or anything, K for the forward reaction, and K off, or that means the K for the backward reaction. They have given us the equilibrium constant, right? And they just want to understand what is the value of the dissociation constant. So this question, though it is from immunology, you really need to know the concept by which you can understand the affinity of antibodies. And later on, this is just a problem of division, a class 4 level operation, right? So it's a class 4 level question, not even a undergrad level question. So this is that easy. Okay, fine. So there is another term known as avidity. So avidity is basically affinity, sum of affinities, and that is valid for only those antigens which has multiple binding sites, such as IgM. For IgM, the total binding affinity for all of these sites, all of these antigen binding groove, is known as avidity. Okay, this avidity depends upon many things: affinity of antigen and the epitope, valency of the antibody, and the structural arrangement of the antibody. Now, when we talked about the antigen-antibody interaction, it is very important to understand the structure of the antigen, because many of the time what happens is the antigen is a 3D structure, right? Now what happens? A particular epitope might be buried deep inside. Let's say it's buried deep inside like this, like my finger here. So, and the antibody would try to detect it, but it cannot detect it because it's buried inside, right? So in that case, you treat these proteins with some denaturing agent such that the thing get opened up and now your epitope is actually released. So why I'm telling these things? Because this question was asked in 2014. See this particular question. So they have asked that a monoclonal antibody against a small peptide derived from a protein X is unable to bind to protein X. So it is actually created against protein X. It should be binding to protein X because it is trained to detect protein X, but it is not detecting. The reason might be, what is the reason? This might be buried inside, right? Buried inside the interior of X. So that is why the interaction is not happening. So today we have looked at so many different concepts that are important for IIT JAM from immunology section. Before we move on further, we should clarify one term which is known as hapten. Okay, hapten is a good carrier. I mean, hapten is a term. Sorry, I just mistakenly said something. Hapten stands for molecules which cannot elicit immune response by their own. That means they are incapable alone, but they are capable when they are interacting with their hapten carriers. So here is our antigen. Now antigens are substances that are specifically, what are the antigens? That are specifically bound to the antibodies or T cell receptors, right? They are either they would be bind to the T cell receptors or they would be bind to the antibodies, membrane bound antibodies. Now immunogens are different. The terminology is different between immunogens and antigens. The antigen or the protein might bind to the receptor but might not elicit or evoke an immune response. If it evokes an immune response, then it's an immunogen. If it doesn't evoke an immune response, then it's an antigen, but it's not an immunogen. So definitely, in order to be immunogen, it has to evoke an immune response, okay? So we talk about a small molecule which might not bind even to the peptide binding groove of the MHC, and that's why they are not displayed. So that's the problem. In order to be displayed on the MHC's peptide binding groove, you need to attain a certain size. A very small molecule cannot be displayed. Okay, so why haptens are not immunogenic? Because of their size, the molecular complexity, etc. So when people injected rabbit with simple haptens, they don't find antibody against that. But while they inject it with the complex carriers, they find specific antibodies against this hapten and carrier adduct. Okay, that means hapten need to be coupled with complex substances in order to elicit an immune response. So hapten terminology is important, and what hapten does, it's important. Okay, so now we move on to understanding what are the procedures used in like in the biomedical research, which involves antigen antibody interactions. Okay, so we focus a little bit on the techniques side. Okay, first technique first, which is most important, enzyme linked immunosorbent assay. You guys already know what is enzyme linked immunosorbent assay, right? Okay, so enzyme linked immunosorbent assay based on the principle that you interact with antigen and antibody and give a color reaction, and that you detect the absorbance of that color reaction to get a idea that how much antigen is present or antibody is present. So both the thing can be actually determined by the ELISA, the amount of antibody and the amount of antigen. So let's see about ELISA. So you have indirect ELISA, whose use is to detect antibody against a particular known antigen. Okay, so first we have a well which is coated with antigen, and I know this antigen binds to a particular antibody. In indirect ELISA, you are looking for that antibody. Your question is, in patient serum that particular antibody is present or not, and you know what is the antigen for that antibody, so you have already coated that in the well. Now you add your annihilate, which is your patient sample, imagining that that has that antibody, so it would bind to that antigen, and then you give a secondary antibody. Antibody would bind to the FC region of the antigen, FC region of the primary antibody. Now if you put some substrates which can be cleaved by the enzyme to give a color product, so it could be detected by your absorbance detectors, and that absorbance would tell you about the concentration of the antibody. So it tells you two things: first, whether the antibody is present; second, if it is present, how much is it present, the concentration range in nanomolars, or let's say nanogram per microliters, etc. First step done. Second step, let's try to look at a live example. So let's say this particular human is affected by HIV virus. Okay, now we're trying to understand that when they're affected by the HIV virus, I mean, what are the possible tests you can perform to understand, okay, this human is affected? It turns out you can take the blood from that patient, it is legitimate, and then you centrifuge the blood and get the plasma and the serum fraction. From the serum fraction, you try to detect antigen or antibody against this virus, right? Now, if you say the antibody against a particular viral antigen is there in the blood, that means the virus has been infected. And if it is not there, the person is not infected, or the virus is not infecting the person. Okay, so what people really do in the lab is do ELISA for the patient sample. So let's say the HIV test would be positive if you develop color. If you don't develop color, the test would be negative. The color is only developing because your patient sample has this particular antibody which is against the viral antigen, which is in green. So this particular antibody is actually coming from the virus, the viral derived antibody. Okay, so this antibody is produced in the host body because the viral peptide was there, and against the viral peptide you have this antibody. So you are able to detect that presence of that antibody with a secondary antibody which is enzyme linked, and that gives you a color. So this is a real hand experience that, okay, how these antigen antibody mediated responses could be useful to detect several aspects of disease, right? We talk about sandwich ELISA right now. So this is a very good method to detect antigen of interest. So you know what antigen is going to bind to what antibody. So you first put a monoclonal antibody against one particular antigen that you want to detect, and then you add your annihilate. Again, your annihilate is your patient sample or blood sample or serum sample or any sample. Then assuming the antibody is present, it would bind, and then you put another enzyme which is secondary, which is another enzyme linked antibody, but this particular second enzyme linked antibody also detect the antigen, as if your antigen is sandwiched between two antibodies. Now this is a monoclonal antibody, the second one, but detects different epitope, so both of that can bind to the same antigen at the same time, and none of that hindered the binding of others because they have their different binding sites and different epitopes. So this is how you can understand whether a particular antigen is present in a sample or not, and the ultimate readout is your color development and getting the color formation and getting the absorbance value. Okay, so comparative ELISA is another particular type of ELISA which is important. The comparative ELISA tells you how much antigen is present, more or less or like that. So let's say you have particular antigen which you pre-incubate with antibodies. Okay, now if you have a lot of antigens, all of your antibodies would be occupied. Now your well also has what? Your well also has a lot of antigen. Now this antigen here comes from the patient sample, and this antigen you have put. Imagining the patient sample has quite a lot of antigen, so all of these preoccupied antibodies would not bind to the antigens present in the plate because those antibodies are preoccupied. Okay, so then when you wash, a low signal would be developed. So now you have a change in signal intensity. If you have a high signal, that means what? Very less amount of antigen is present. That's why most of the antibodies were free. It can bind to the antigens, and as a result it can give you a dark color. But if you have a light color, that means quite a lot of antigen is there. Okay, fine. Now we look at pregnancy test. It's an important question which was asked in 2014, and this time IIT Kanpur is doing the session. The IIT Kanpur is doing the JAM test, so probably it's an important concept. So in the pregnancy strip you see two bands and you see, okay, I'm pregnant. How does it happen? So what does pregnant, I mean positive, negative or invalid means? It's based on the band, so it would be really clear at the end of this video. So you put your urine sample, I mean you means a pregnant woman who is suspected to be a pregnant. She put her urine sample, let this urine like really go or spread through a capillary action. Now in the annihilate zone there are specific regions. Ultimate readout is two bands, and the woman would think, okay, I'm pregnant or not based on the presence of these bands or not. Let's see how these bands are formed and what you're detecting. So you have your reaction zone, you have your test zone, and you have your control zones. All of these things has significance, but let's see what we are detecting. When the embryo is implanted, one particular hormone is secreted by early placenta known as human chorionic gonadotropin. Human chorionic gonadotropin helps in many things. First of all, human chorionic gonadotropin makes the corpus luteum more or allow the stability of the corpus luteum, or ensure the corpus luteum don't degenerate and becomes corpus albicans. So that what happens is like the corpus luteum can secrete always progesterone. Progesterone can ensure that endometrial lining is pretty thick and during the pregnancy it does not collapse. So moral of the story, a pregnant woman's urine would have a little bit of HCG, but a woman who is not pregnant should not have any HCG present in her urine. So that's the key point. Now let's say a pregnant woman has put her urine on the strip. So as the urine flows, what would happen? These antigen would bind, these antigens which are present in the urine, which is actually human chorionic gonadotropin, binds to these antibodies. As the urine flows, these antibodies would also move out to the further region, the test region and the control region. Now in the test region, you have already anti-human chorionic gonadotropin antibody. Again, these are monoclonal antibody but against different epitopes. Previously in the annihilate region, you also have these monoclonal antibodies which are anti-HCG but with different epitopes. Here you have HCG antibody and you have some cleavable dye molecules. Now as the antigens, as the antibodies bound to these things fall, get to that region, what happens is they bind to these human chorionic gonadotropin bound antibodies, right? And the antibodies here are already bound to enzyme, so they can cleave the dye and they can give the first band. That means if chorionic gonadotropin is present, it is going to be detected and going to give me the first band. What about the second band? Let's look at it. The second band is found when you have a mouse antibody. Let's say this particular antibody, which is human chorionic gonadotropin detector, but it is actually raised in mouse. So this particular mouse antibody would be detected by an anti-mouse antibody, and if it is detected, while it is flowing through the urine, so what is going to happen? The FC region is going to be detected by the anti-mouse antibody. As a result, what would happen is a cleavage of this dye molecule by these enzymes, and there would be a second band. Now this explains the control band. Regardless you have the human chorionic gonadotropin or not, you would always see the second band, because the antibodies which are present in the test zone where you drop the urine, these antibodies should be binding to these particular anti-mouse or anti-rabbit or anti-species antibody where these are actually generated. That means two band means the woman is pregnant. One band, you see the test band, don't see the control band, then that means something is wrong with the test itself. But you should always go to the doctor and check up, because there could be cases where many cancer lead to secretion of human chorionic gonadotropin into the urine. So in this whole video, we have really covered a hell lot about B cell development, about B cell maturation, antigen antibody interactions. We looked at questions, important topics. We looked at how antigen antibody interaction is used in several immunological techniques, etc. So I hope this 1.5 hour of time was productive to you. Even if you are watching the repeat podcast, at least you would be able to get some marks out of it in IIT JAM. So with that, I would say all the best and I would sign off. But I would always let you know that I am also present in Unacademy Plus platform, where the lectures are more relaxed. This is like pretty much bullet train like, but I don't have any option. I have to cover a huge spread in a span of 1.5 hour. So I hope you enjoyed this video. You can always slow the video speed to watch this video. So see you next time. Thank you.